The broadcast is now starting. All attendees are in listen-only mode.
Welcome to today's webinar on the update of for vascular products and their impact with the new ISO 10993-1. My name is Thor Rollins. I'm our Vice President over medical devices here at Nelson Laboratories, but my background is in biocompatibility. I've been with Nelson for almost 25 years now, and in that time, about 15 of those 25 years, I've been involved in the ISO committee. I am right now the co-chair of Working Group 1 for AMI, which is the working group that works on the ISO 10993 Part 1. So I'm very familiar with the changes. And my whole background and experience is in biocompatibility. Preston?
Yeah, Preston Zook here. I'm a business development manager within our sales organization. I've been at Nelson Labs for 17 years, and for over 10 of those years, I worked in our particulate lab. So I was on the working group for TIR 42 to help it get published to the 2021 version, which will be discussed later. Although I work in our sales group now, I come from a pretty long stint in our particulate group.
Great, thank you, Preston. And in today's webinar, we're going to cover a little bit on the updates to the new 10993 that should be published any day now. We check every morning. It literally can be while we're doing this presentation. So we're going to talk a little bit about the biggest, what I think are the biggest impacts to the community. We have gone into more details on these impacts in previous webinars. So feel free to reference those if you're looking for a more in-depth to changes of 10993. In this particular webinar, we're going to kind of focus a little bit more on what could be impacting vascular products, and then also a little bit more in depth on some particulate concerns, which we're seeing not just from regulatory bodies, but also from this standard update. So jumping right in, I have a timeline here of the standard, and the one thing I want to kind of just point here is that by the time that the ISO committee saw the first draft of this standard was July of 2023. And so it's been a little bit over 2 years, which is a very, very short time to have a brand new standard come out and be approved. And so that's been honestly a little bit difficult for a lot of us working on the standard. There's been significant updates. In fact, it's almost a completely new standard. And so there's been a lot of updates for a year and a half to 2 years of work. And so we're going to talk through what that means as far as acceptance goes, but also kind of the things that were probably not mature yet as far as concepts, but we're still going to have to deal with it. Okay, so what does this update mean specifically to the vascular device industry? This is going to be also impactful to almost all types of devices. But specifically looking at vascular medical products, that the main impact is going to be an additional endpoint to be evaluated. So if, regardless of contact type to the body, the medical device, the vascular products will have to look at genotoxicity. And we'll talk a little bit more about that here in a second. We're also going to talk about some issues with foreseeable misuse, which will kind of focus on vascular kind of contact and what some foreseeable misuse might be. There's some new guidance about cumulative contact duration, so we'll talk about that. End of life evaluation, and then Preston's going to jump into what particulate evaluation might look like for vascular devices, and I'll talk a little bit specifically about what the new standard means as far as enforcing or requiring more of that. So this new standard, I just think it's important that everyone should know that this is the very first committee vote on the first draft that you saw in July of 2023. As you can see, Germany and US voted no. Everybody else voted either yes or abstained. The one consistent for every single vote that happened, and that was every committee draft, every final draft, draft international standard, everything that had a vote, US has voted no every single time. So we're still a little uncertain exactly how the FDA is going to deal with this new standard. I am almost sure that it's going to be a partial recognition. The things we're talking about today, I think might still be enforced. There are definitely some that are already being enforced, like end of life and particulate, and the cumulative use duration, and the genotoxicity, I guess. So pretty much everything we're going to be talking today, the FDA is going to enforce or recognize. But I do expect partial recognition, and there's reasons behind that. If you want to know why, you can go back and listen to some of our other webinars we did on this topic. So what's in this, and we call it FDIS now. For those of you that might not know AMI or ISO nomenclature, FDIS stands for Final Draft International Standard. That's what it's kind of right now it is, but it was accepted about 8 weeks ago. So it's going to be the new standard just as soon as it gets published, like I said, any hour. But what's in this? So this first one, which I kind of already talked about, which is increased genotoxicity evaluation. In the past, for vascular products, if you have limited contact with the vascular system, so like you think about delivery device for a stent, or any kind of like delivery device or external communicating device that had maybe just less than 24-hour contact with the vascular system, you weren't really required to assess genotoxicity. The standard today, and this is the new table for contact with blood, as you can see, any contact, regardless of time of contact with circulating blood, will require genotoxicity evaluation. So for those people who have, like I said, in types of catheters or delivery devices that have less than 24-hour contact, this would be a new evaluation for you. And so you have to consider genotoxicity going forward for these types of devices. So keep that in mind when you start to look at new devices or changes that are going to the FDA or your notified bodies in the next, well, basically from this point on, because the standard's going to be out any day now, you're going to be required to do genotoxicity. The next kind of change to the device or to the standard that honestly is probably one of the most controversial, there's been a lot of argument and discussion, is about foreseeable misuse. Now, one of the things why we rewrote this standard was to align it with 14971, the risk assessment standard for medical devices. In there's a whole concept about foreseeable misuse. You have to evaluate how your device might be misused and have, what kind of impact that could have on the patient. We now have taken that concept and put it into a biocompatibility concept. So when you start looking at things that could be foreseeable misuse, this is things like, body weights or, populations or length of time, things like that. So for example, if your catheter or vascular device could be used in a patient population that's smaller, like children or even neonates, even if you contraindicate not to use for that population, if it could be used and reasonably be used in that population, you still have to evaluate what kind of potential impact from a toxicological standpoint that could have to those type of patients. That also means for length of time. So if you think that the typical dwell time for your vascular product is 8 hours, but it's foreseeable that someone could use it for longer, longer than 24, then you still have to evaluate the endpoints for prolonged contact. So these are things that I think understandably could be looked at as biocompatibility concerns, but the concern from a medical device manufacturer's point of view is now you're relying on what your regulatory body, be it notified body or FDA, sees as foreseeable misuse. And so you guys might not see it as a foreseeable misuse, but they may interpret it as more of a risk. So communication with your notified body or FDA or any regulatory body is probably pretty important here. The third thing we're going to talk about is exposure duration. So there wasn't much guidance in the past in 10993 about it. It just talked about less than 24 hours is limited, 24 hours to 30 days is prolonged, anything over 30 days is long-term contact. So most people, let's say you had a delivery device for a stent, or let's say you had some kind of catheter that was going to be in contact with the vascular system for, we'll use this real life example here in the standard where for 5 minutes. And you can think worst case, they could do that once a day for a week. And so most people would say 5 minutes times 7, that's 7 days in a week, and they would say 35-minute contact. And they would say, okay, that's limited contact. The majority of our clients did it that way. The standard now says that you can't do that. So basically, regardless of time, one contact is a day. So in this scenario where you have everyday contact for 7 days, it would be 7 days contact. And so it would be a prolonged contacting device. And the reasoning behind that is even though the device might only have 5-minute contact, the chemistry being left behind in the vascular system is now being dealt with the body throughout that day. And we don't know if it actually, is metabolized and excreted or dealt with, or if it starts to bioaccumulate over time. And so that's why, unless you can prove that it's not bioaccumulating or being, metabolized within 24 hours, all your chemistry, then you have to consider it a day contact, because that's how we deal with things in toxicological concerns. So this is going to change a lot of devices' contact time, because like I said in the past, they've really just added up the time actually contacting the body and dealt with it that way. By the way, this was brought up by the FDA, so I'm pretty sure this is going to be recognized. This portion will be recognized by the FDA. Okay, the other kind of big thing that's changing is end of life. And for vascular devices, this most of the time means shelf life. What could change for their polymer as it sits on the shelf, but not always. I know of reprocessed vascular devices. I also know of single-use devices that then are reprocessed by third parties that also fall into this area. And the concept here is most people do biocompatibility at time 0, which honestly is the right thing to do, because you have most of your volatile and maybe even semi-volatile compounds at time 0 that interact with the body. But especially polymers and other types of materials like that can change over time. So you have like, stabilizers, and you have, other additives to polymers that are meant there to kind of help with, different types of chemical degradations and things like that. But those break down, so you'll start to have blooming, or you start to have changes of surface characteristics, or even leachable profiles. And so the concept now is you have to evaluate your biocompatibility just not at time 0, but at the end of shelf life, and potentially in between, depending on your materials. We're not saying you have to redo all your biocompatibility. You have to just do some kind of evaluation at different endpoints in your life cycle to make sure that this device is not going to change its biocompatibility impact to the patient. So we're still kind of determining how this is going to be enforced, but this is something we're seeing today. So in fact, in the last month, I've dealt with 15 different responses to the FDA about end-of-life evaluation. So if you're not seeing it yet, you will be seeing it soon from FDA and once the standard's out, notified bodies. So one of the other things that this standard is bringing in specifically for devices where this is a potential risk, vascular devices definitely falls in there, is particulates from your device. And so when you start looking at different type of biological exposures where particulates could be a concern, orthopedics could be a concern for particulates for sure, especially in joint spaces. But one of the bigger ones are neuro and vascular. And so we're starting to see this a lot from the FDA already. So in fact, I just got done talking to a company that has a bunch of vascular devices where all those vascular devices have been asked by the FDA to look at particulates because they're worried about shedding of particulates into the body. Now, from a biocompatibility process, we have, and Preston's going to talk about this in a second, we have some good test protocols set up to look at cardiovascular devices and measure the particulates coming out. What we don't really have great for medical devices is acceptance criteria. In USP, we do have a section where it talks about acceptance criteria for pharmaceutical containers. And so we use that today to kind of help, say, hey, if it's good for injecting fluid into your body, drugs, then it should be good for medical devices also in the vascular system. But right now, Nelson is actually preparing so we have decades of data for medical devices on particulates. And so we're right now using some AI and some help to take all our data and kind of do a S-curve on it, trying to get a normative distribution to try to figure out, obviously these devices are safe because they're on the market and they're not causing, thrombosis or embolisms or things like that. So can we try to use our data to kind of recommend some medical device specific safety requirements based on how the device is used and things like that. So we're currently working on that. We're hoping to publish that next year, but right now we still use the USP acceptance criteria. So right now I'm going to turn the time over to Preston to get into a little bit more about particulate testing and how we do it here at Nelson Labs.
Okay, thanks, Thor. Yeah, so here, this slide kind of goes over two primary considerations for particulate matter testing on devices. One is what Thor sort of alluded to, which is established compendia. So that's something like USP Chapter 788, ISO 14708 for active implantable devices, something like a lead or a pacemaker, 8536, 1135-4. And then also the other side of this is TIR 42. So we're going to, compare these at sort of a high level to help understand why it's important for vascular device companies to test according to TIR 42. So a quick snapshot below is, basically showing that an established method, for instance, 788, that's sort of like, the most well-known standard for particulate matter testing. It doesn't provide any guidance for how to extract a device, but it does provide guidelines for what particles to assess, what particle sizes, and then it does give acceptable limits as well. If we shift over to TIR 42, this encompasses a very wide variety of vascular devices. So catheters, delivery systems, vascular imaging devices, embolism devices, and it allows for a framework that if you have a vascular device, you can create a test system and dive a little bit deeper into the particle shed and what that means for your product. So if we go to the next slide here, again, this is sort of a snapshot to help provide a little bit of reason as to why moving outside of method compendia and into TIR 42 for a vascular device is important. So TIR 42 still allows for a lot of customization when it comes to your test method. So, again, for something that's referencing 788, with no guidance for testing a device, there's not a lot of framework for you to put into place to test your vascular device. With TIR 42, you're leaning on the IFU, you're leaning on clinical applications, and then not only are you just getting an output of the number of particles in a given size range, you can take it further. So an easy example of that, to begin to look at what you can assess in terms of device safety, is particle sizes. So in the images below, the annotations there are small, but the larger particles, so the one on the left and the one on the right, those are measured at almost 200 microns and over 300 microns. In a standard USP 788 reference test, that's going to show up as one particle greater than or equal to 25 micron. But with TIR 42, you go one step further or even beyond that to sort of understand what's shedding from your device. Is this material from your vascular device system that's shedding because it's not entirely compatible? Are you having coding integrity issues? And what does that mean on the back end for an end user? So this is a TIR 42 2021 version overview and how it applies. Another thing to note is that the prior version of TIR 42 was in 2010, and so the 2021 version was a complete overhaul. There's a lot more information. It provides guidance on, particle ranges to assess, so not just 10 and 25 micron, but 10 micron all the way up to, 500 or 1,000 plus micron. It also introduces both methods of analysis, so light obscuration, which is the automated method, as well as light microscopy, which is the manual method. Both have their own sort of pros and cons and inherent flaws, if you will. And then it also goes beyond just sizing and enumeration and starts to look into chemical makeup of particulate using something like FTIR. So the general outline for a vascular device test using simulated use can be broken up into 4 steps. So the first is a model validation. First, you have to define your test system. So an anatomical model is designed usually using worst case tortuosity as it relates to your patient population. It's also using appropriate materials, so something like glass. And then once you have that designed and you have introduced your ancillary test components, so guide wires, dilators, things like that, you would do a spike in recovery efficiency test. So we introduce a known concentration of particles at a known size. We will run through what would align with the IFU in the test, and you evaluate how many particles you were able to recover. So TIR 42 provides guidelines on percentages and acceptable parameters for the model validation. Once your model is validated, you would go on to the device extraction. This is a process where we tailor the IFU to a particle test and essentially just run through a worst case scenario. How many times can this device be inserted, reinserted before deploying a stent? Those considerations should be built into your device extraction, but for the most part, this is heavily based off of the IFU.
And Preston, just to add, that's one of the things where this foreseeable misuse might go in, because like Preston said, it's heavily based on the IFU, which is what we recommend. But one of the fears I always have is when you start seeing a foreseeable misuse, because one of the things that the regulatory bodies always said is they see some companies try to protect themselves by putting it into the IFU, but they know that the company is that the doctors are using it off-label or not following the IFU, but the company refuses to change IFUs because they don't want to test for it or things like that. So this might be one area where even if your IFU says something, but people are off-label using or not following your IFU, you still might want to consider setting up your device extraction based on clinical evidence. But I think that's up to every single company to kind of think about.
Thanks, Laura. And then another thing I wanted to highlight on that step specifically is testing against a predicate device. So while USP Chapter 788, for instance, has acceptable limits, it's most common to test against a predicate device because otherwise you're sort of bound to that 6,600 particle limit that's built within USP, when in reality, a much higher number or a much lower number might be more applicable. So for large delivery systems, things with many ancillary components that are interacting and there's friction going on in particle release, you may not want to compare that against 788. You'd want to compare that against a predicate. If something's already on market and justified as safe, then you might set yourself up for success to test against a predicate. Something like sample size is not directly detailed in TIR 42, so sample size justification should be prepared as well. So we have the model validation, we've moved on to device extraction, and then the third portion is particle analysis. So again, we're typically using both our methods of analysis to look at particles. So light obscuration for smaller particle size ranges, so that would be something like 10, 25, and 50 micron. The light obscuration method is typically better at accurately sizing those smaller particle sizes because they remain homogeneous in a suspension. It's highly sensitive, but moving over to the microscopic method, that's typically a better way to make sure you're capturing all of those larger, more dense particles that during analysis, if you're using something like a HIAC system, could settle towards the bottom of a collection container. So the microscopic method allows you to capture for those larger particles. And then you can go beyond that by performing some imaging, like in the last slide. And at that point, you've isolated the particles on a slide such that you can move on to further identification, so something like FTIR or SEM. And then another pretty big consideration in TIR42 is just overall device performance as it relates to something like coding integrity and just overall degradation of a device through use. So, you know, you could learn that running through your IFU is actually not working well with something like a new coating manufacturer change. So what we can do is we can run through a simulated use after we have initially dyed the stain, dyed the coating using a stain. And then when we have run through the simulated use procedure, we can then inspect the devices in magnification or just with the naked eye and look for, you know, chunks of coating missing or just any sort of degradation. And that can be a pretty telling portion of the study. If you have high particle counts, you know, maybe you're seeing a poor performance from the vascular device system that is, you know, sort of damaging. So that sort of summarizes the overall process for a TIR42 study. And, you know, it helps showcase why it's important to investigate beyond what method compendia offers or the lack thereof. And it really allows you to tailor a device test plan specifically for your needs. So thank you for watching. You can reach out with any questions or follow up. We're happy to have conversations and discuss any things you might have going on. Here's some of our contact information. And then, of course, all of our webinars or other learning tools are on the Sotera Health Academy that you can find online anytime.
And I want to thank everyone for taking some time and listening to Preston and I, and learning a little bit about how the new 10993 standard is going to be changing and potentially impacting, especially around vascular devices. And like Preston said, please reach out to us with any questions. We're always available and be happy to answer anything that you guys have around biocompatibility or particulates. So thank you for your time.